Good afternoon, everyone. I want to welcome you to today's webinar. Before we get started, I want to go over a few housekeeping rules. Please, everyone, mute your line. You can use the chat box to ask any questions, and we'll be answering all questions at the end of the webinar. For any additional questions that may come up and are not answered during the webinar, you can email us at education@nola.gov. And this webinar is not offering CEUs. The next voice you'll hear is our director, Dr. Claudia Rigo, who will introduce today's speaker.
Thank you so much, Astacia. Thank you, everybody, for joining. Thank you also to Dr. Jameson to be here with us today. So Sam is a senior postdoc fellow at Tulane University in the laboratory of Dr. Don Wesson. He completed his PhD in 2017 in Dr. Wesson's lab. After 3 years of postdoctoral research at the University of Mississippi Medical Center in Jackson, we're so happy that he returned back to Dr. Wesson's lab for further training in vector biology and vector ecology. He has studied populations of Triatoma sanguisuga in the southeastern Louisiana since 2011. So we're super excited. He's actually going to be presenting, this paper just came out, I think earlier this year. So he's going to be giving a really great review of some of the research that he's done. So Sam, thanks so much for joining us today at our webinar, and thank you everybody for attending. Sam, take it away.
Thanks, Claudia. Sorry, everybody. I was out sick 100% yesterday, and I'm only up to like 80% functional today. But we will make it through this. Let's see. There we go. All right. So thanks everybody for tuning in. I know these things get often put in your mailbox at the last minute, so happy to see so many people here. I am talking about Chagas in the Southeastern United States today. It's a big topic. It was a big review haul that we did. Came out a couple months ago, and so I'm just going to try to focus in on a few aspects of it that are hopefully more relevant to the community, the vector control community, and Bateman districts and people who work in public health around vector-borne diseases. So just going to go through some quick background, make sure everybody is familiar with the system that we're talking about, because this is outside the realm of typical mosquito-borne diseases or other, other vector systems. It's kind of its own little area of vector-borne disease. I'll talk about the regional insect vectors and parasites, and look at a couple quick case report type things that we have seen over the years here in the Wesson lab, and then touch on the veterinary health and human health implications. So just quick background, overly simplifying this system. So Chagas disease is caused by infection with the parasite Trypanosoma cruzi. Trypanosoma cruzi is a single-celled kinetoplastid protozoan parasite. So that just means it's a single-celled animal type thing. It doesn't have a sexual reproduction stage that's well defined in any way. It mostly reproduces just by splitting itself. It's a kinetoplastid protozoan, which just means it has this extra organelle called a kinetoplast. And it's something that we use to group protozoans since we don't really know what they are properly. So it is in the same group of parasites and is closely related to the African trypanosomes that cause sleeping sickness. And those are so this is American trypanosomiasis, and that's African trypanosomiasis. Many true bugs, so hemipterans in the subfamily Triatominae, are able to transmit this parasite. Again, this is a American, North and South American type vector system, so it is almost entirely contained in this hemisphere. And so true bugs over here that evolved from assassin bugs are the ones that spread it. And T. cruzi is thought to be much older than its insect vector system. And so this is important because a lot of times when we talk about parasite vector systems, we think that they've always been this way and that they're perfect systems. And the consensus right now is that we are actively watching the evolution of this vector system. That Trypanosoma cruzi has been around since South America and Africa separated, and one group became the African trypanosomes and one group became the American trypanosomes. And that it cycled between animals without a vector for a long time. And that the Tritomine species that spread this parasite have evolutionarily recently picked up this habit of spreading the parasite. And so it is not a perfect system. They're not great blood feeders. A lot of these bugs can actually subsist on non-blood hemolymph type things if you just feed them other insects. And so they don't like require blood, a lot of them, in the way that mosquitoes do that we work with as vectors. So it is kind of a wonky, imperfect system. Now, there are a few species that are super highly adapted to humans, blood feed great, really need human blood or vertebrate blood. And those are kind of the exceptions in this kissing bug family of vectors. So both the parasite and the insect vectors are native to Louisiana, to the Southeast, to this region. This is not exotic. This is not new. This is not imported or invasive in any way. This has been here forever. We're just really starting to look at it. People have probably been getting Chagas disease in the Southeast for as long as people have been in the Southeast. So it's more just about looking closer and more fully characterizing what the issue is. So this is the life cycle of Trypanosoma cruzi. So if we start at the top, I don't know if my cursor will show up. So we start at the top, the kissing bug at 1, the bug takes a blood meal, and this is the classical pathway. It excretes the parasite in its feces next to the bite while it's feeding, and the bite is painless. But after the bug walks away, there's some irritation in the bite, and maybe you feel the bug on you, and so it feeds when you're sleeping. And so when you push on it and rub on it, you accidentally rub the parasite into the open wound, and that's enough to establish an infection. The infectious dose for this parasite is 1 parasite. So when we talk about viruses and lots of either other, small microorganisms that are pathogens, we talk about the infectious dose needed to establish an actual infection. It's usually some significant number because your body's pretty good at weeding these things out. The infectious dose, though, for T. cruzi is a single parasite, is all you need to get into your bloodstream to establish a patent infection. So this highly infectious stage that gets passed in the feces is called the metacyclic trypomastogote. It can cross intact mucosal membranes. So you'll see people who get infected in their eyes by pushing feces into their eye. It can infect the mucosal membranes of your mouth. And even if there's no opening, just being intact, it can cross those membranes. Gets into circulation, invades cells, and becomes this amastogote stage where it replicates by binary fission, just racks up the numbers, and then it lyses the cell, goes back into circulation, and repeats this cycle. At some point, your immune system kind of tamps down the circulating parasites, and they live more in this amastogote stage and are slower growing. And so you don't have so much parasitemia circulating. And then this infection goes on for a long time. Some people spontaneously clear, most people hold on to it for the rest of their life. And a good portion, the majority of people who are infected never have any symptoms. They die of something else before this ever becomes a problem. But if it does become a problem, it causes often either gastrointestinal, like esophageal megaesophagus or megacolon, or it can cause issues in the heart lining. And while the parasites are circulating, the bug side of the transmission cycle is they just pick it up in a blood meal, and then it just divides by binary fission in their stomach. This is a far less complex, it's a stercorarial parasite transmission route. So in the poop, And it is far less efficient than a mosquito bite. Think of something like West Nile that gets injected when the mosquito bites from their saliva. This is far less efficient and it's far less complex because the parasite never has to infect the bug properly. It never jumps into the bug or invades the bug's tissues or anything. It just grows in its gut and gets passed out the back end. You see the little raccoon and the dog and the mouse down here. So these are well-known other places that the parasites are found and good hosts that we'll get to in just a little bit. So our regional parasites, which is not something that is often considered unless you think a lot about the life and times of an individual parasite. But what we consider to be T. cruzi is actually a lot of different types of organisms that are all the same species as far as we know, but we have these different lineages, which are T. cruzi 1 through 6 that you see across the top, and then TC bat, which is aptly named because it lives in bats. But it is a Trypanosoma cruzi. It is the same species. For a long time, we thought that T. cruzi 1 and T. cruzi 4, as you can see up here in the light blue in the US, were the only two that lived up here. And that there were these big geographic differences in where the different lineages of Trypanosoma cruzi lived. And that has recently been changed because now we know that in the Southeast, T. cruzi 1, T. cruzi 2, T. cruzi 4, T. cruzi 5, T. cruzi 6 all live in the Southeast for sure. We suspect T. cruzi 3 is probably here too. We just haven't come across it yet. Dr. Dumonte in our department here has done a lot of work with this with Claudia Herrera in trying to tease apart non-typical T. cruzis that seem out of place. And this is a lot of deep sequencing work that they've done from primary collections here in Louisiana and related and close states. So we do have these T. cruzis. T. cruzi 4, 5, and 6 are thought to cause more megaesophagus, megacolon symptoms. But again, all of this dynamic is kind of changing. Yes, for people in the US that we see, we have cardiomyopathy is the major clinical symptom that we would see, but we have seen at least one case where there was a llama that was infected that had mega syndromes in the gastrointestinal tract, and that was in Louisiana. So our regional vectors, this is the part that I like. I'm a vector person. So we have Tritoma sanguisuga and Tritoma reticularia. So this is the current slide, and it is not updated yet. This is the current, this hot off the CDC's website. They haven't updated the name for Tritoma reticularia. It got reclassified at the genus level recently, and so it's actually a Paratritoma reticularia now. Excuse me. So these are the two that are in this region. You can also see that on the reticularia map, Louisiana is missing. We don't know if it lives here. There's one potentially believable collection from Western Louisiana back in the day. But that's something that we are looking into, trying to figure out if it's here for sure, and if it's not, why, because it does live all around us. They're very similar looking, and they behave differently. Sanguisuga is the one that we're going to talk about for the rest of the time. So Tritoma sanguisuga is the major vector in the eastern United States. It's the eastern blood-sucking cone nose is its common name. So this is it on a quarter. So they're hefty bugs. They take a good bit of blood. They're this bright orange manded assassin bug looking bug. So it is, they have a rostrum tucked up underneath their head, and that lies back underneath their thorax. And so these two bugs look almost identical when you look at them just from the top. Reticularia tends to be a little duller, but that's never something you want to really go by. Its head is a little shorter and stouter than sanguisuga. And for the longest time, the definitive separation between these two was putting them under a microscope and looking for setae on the rostrum, because Paratritoma leucicularia is a fuzzy little bug when you look at it under the microscope, and sanguisuga is not. But now, thanks to a lot of hard work by a lot of people, it has been convincingly shown that these are actually in separate genera. And so now we have this whole big, great question about why they look so similar, if they're so genetically disparate, if there's some convergent evolution reason for them to look like this. The back ends are differently shaped, but that's a male-female dimorphic thing. That's not a species difference. So this is a female with a pointy abdomen, and this is a rounded abdomen, then a male. And you'll find these all over wooded areas of Louisiana. So they're not going to be in urban areas. They're not even going to be in suburban areas unless they have big old growth wood stands close by. Sometimes you kind of get one-off drops of bugs flying in. They have about one good flight in their lifetime where they can fly 100 yards or more in search of, we don't know what, we assume a new place to live. And so you'll get those sometimes in suburban areas. I know in central Mississippi, we got some in one of the bigger suburbs outside of Jackson, but it was right next to an old growth stand of woods, and you could see how the bug just kind of hopped in. Similarly, we had one in downtown Jackson, but it was at the edge of an old growth hardwood forest that's maintained as a national or a state park. So undisturbed woods is really what you'd be looking for. So our primary case, they got the Wesson lab involved in all of this, was the case that I'm sure many of you are familiar with, which was the 2006 case of locally acquired human Chagas disease in Orleans Parish. So that case, the home has been extensively sampled since then. I know I got my dissertation off of samples from there. I assume other people have as well. That we're still collecting. So that's been, it's well over a decade at this point that we've been collecting annually from this place. And basically, there were 2 residents that lived there full time. Only one of them ever became positive with human Chagas disease. The other one has been extensively tested, as you would imagine. It's never seroconverted, never shown any signs of infection. Both residents had multiple bites and multiple risk factors. So this is not something that, and they live in a very rural, wooded, old growth area of like rural Orleans Parish. So this is not something that, it made big news because it was like, oh no, it's in New Orleans. It's not downtown. It's not in anywhere where there's any concrete around. And the risk factors involve things like sleeping uncovered outside, like without a tent or any kind of protection, not having air conditioners in your house, having self-made construction for your home. So, non-modern construction methods that allow for cracks and crevices and big holes kind of to the outside that a bug could squeeze through. So they were, the residents were living their life just like they wanted to in a house that they built largely themselves, and loved to be out and in nature. And they had a large amount of bug exposure and bug bite exposure, and still only one of them ever converted. So again, this is a super inefficient transmission system, but it is something that exists and it does live here. So it is something that we should think about and be on the look for, and probably exclude bugs from around houses as much as possible. We have effectively controlled the other arm of us going out and collecting bugs at this place for years and years on end has been that we've consulted with them on a commercial applicator, with them and a commercial applicator, to spray the house to try to figure out what works best to control these bugs. They don't have any kind of grooming habits, so residual sprays are less effective than you would hope that they would be with something like a roach. But they do work. So we do monthly sprays, or we recommend, and they do monthly sprays of the property, indoor and outdoor residual spraying with the highest label rate of whatever they're using as the cockroach residual spray at the time. That seems to work. Because again, they have like one good flight in them. They're not super mobile bugs. So they kind of hit the outside wall when they fly in, have poison contact, make it inside, have more poison contact. And we have them running air conditioners now. So most of the bugs that we find are heavily poisoned and within the first, 6 feet inside of the residence or the wall. So it's been very effective. The biggest thing, though, for control there was an environmental control that we accidentally had happen. So we had no control. It was a totally uncontrolled accidental experiment. But there was a construction on the property that was taking place, a large, like actual new building being put in with a foundation and everything. And what they did was they brought an excavator to clear out some of the old growth woods that were there. And the quote from the construction worker was that he had never seen so many rats in his life. And so apparently what they had disrupted was a multi-generational large pack rat midden that was constructed there and pretty well camouflaged. It looked kind of like a hill, but now that it's gone, it's clear that it was built up, that the ground is actually flat there. And when that happened, we went from collecting annually about 100 bugs a season to 0 to 1-ish per season. This year we got 3 bugs, and that's kind of been a boon here in recent years. So again, these are only adults because these are the ones with the wings that can fly. So the nymphal stages do not have wings and kind of stay put in their nest. My running theory, we're still trying to tease this apart, this is not in a textbook, is that they probably are holed up with a wood rat or a pack rat. Neotoma floridana is probably the species that they're most closely related to. But it could be a rice rat or something else that has a multi-generational house, just because they're such long-lived bugs. They need stable housing for like more than a year to make it from egg to adulthood. So that brings us to the second case that has informed a lot of what we think about Tritoma sanguisuga, which was a beehive out in Bayou Sauvage or Bayou Sauvage adjacent. So one of the botany faculty from Uptown flagged old set of old beehives, and he said, "Oh, I think if I remember right from entomology in graduate school, this was a kissing bug. So y'all should go out and look at it." And he was right, which is good because the nymphal kissing bugs are not nearly as brightly colored as the adults. They're kind of dusty orange, and they cover themselves intentionally in dirt to camouflage themselves. And so his eagle eye spotted these bugs in the bottom of a abandoned beehive. And we turned over the beehive, and there was a pack rat nest. We never saw the rat, but it had all of what you would imagine in a pack rat nest, little pieces of scrap and plastic and shiny stuff. And it was out in the woods. It was out like not around people. So it didn't look like there was some trash around it. It looked like shiny trash had been brought in with it. So it looked like a pack rat nest, but we never did the genetics. Didn't take the samples at the time to figure out what the actual species was. But there were hundreds of these bugs living in this tiny space under a beehive, and they were all aggregated right up on top of each other. And we removed the cover, the whole beehive cover, because they were under the base. And so, of course, the rat never came back. And we went back and did weekly collections for 4 weeks, and were able to recover bugs at each collection. And then we kind of looked in circles around the where the beehive was, and we never saw any bugs that were leaving or dispersing from the area. So this has really informed our ideas about what this bug probably does in nature, that what we see at people's homes, the adults, and sometimes even nymphs that are around people's houses, um, are incidental bugs. That the real lifespan, the natural history of the bug is probably much more tightly associated with rodent in a nest where there's an easy food source that's stable, and they can have generation after generation of bugs right there. And then they would just kind of incidentally lose some that go out as they get older to establish more nests. So it does help us because it would hint that if we could figure out exactly where they're living in the woods, then we could target control efforts away from a house. And that would let us, not have to apply a bunch of residual sprays at the highest legal label rate in and outside people's houses, which they appreciate, and I'm sure the people who apply it appreciate. But that we could treat the nest and really tamp down the population if we're actually cutting out the stable kind of population sink out in the woods. So why do we care? Are we just hunting this bug for no reason? Is it just because it's cool, but it has a rare disease? And yeah, that's pretty much what it is. So it does feed on humans. So Tritoma sanguisuga is the most often implicated vector of locally acquired Chagas disease in the US. So that includes the Southwest, where there are many more kissing bug species, Texas, which has many more kissing bug species. The most often, when you get a human bite exposure, bug was found in house, or a certain species was implicated in a known locally acquired Chagas case, almost all of those cases involve Tritoma sanguisuga. It seems to be the one with the least fear of humans. It will sneak up and feed on a person. There was a case report from, I think, South Carolina, where a lady was sitting out on her porch and a bug came up over like the back of her chair and just bit the back of her arm while she was just sitting out, just middle of the day. So they do aggressively feed when they're hungry. They'll feed on just about anything. There are reports of them feeding on, of course, roaches and other insects because they are assassin bugs at heart. But they feed a lot. When we do blood meal analysis, we get a lot of frog, a lot of like a knoll lizard. So it's kind of whatever's around that they'll feed on. But for each molt in their lifespan, they do require a vertebrate blood meal and to get to the next stage. And so this species is a peridomestic or sylvatic species. We're still trying to tease apart which category it more properly falls in. There are parts of its range, like in Florida, where they see more peridomestic activity in entomological surveys. In Louisiana, it's basically a sylvatic species that has incidental kind of peridomestic fly-in encounters with humans. So it really does prefer to be out in the woods and not right up next to people. We've never seen it colonize a house, even in areas like Florida, where they report more peridomestic activity and bugs going in and out of houses, bugs of this species. And they sometimes do find nymphs in the house. No one has ever shown that Tritoma sanguisuga can successfully colonize a house, an actual human dwelling. And that's in stark comparison to South American species, who are the major drivers and efficient vectors of this human disease. They readily colonize houses and will live right up next to people. And so if you look in medical textbooks, a lot of you'll get these pictures of like a thatched roof house, and the bugs will live in the thatch, or they'll live in the walls, cracks in the walls, and they'll come out just like bed bugs and feed on people at night and go back into the walls. We don't have that here. thankfully. So these aren't like a impending threat to humans that we know of, but they do incidentally cause some pretty awful disease in people. And it is a system that isn't well understood. So we're trying to understand it better, see if we can tamp down some of that. The other problem with Trichomonas sanguisuga is that they have high rates of T. cruzi infection, at least in Louisiana. Most other places that they've looked, that's also the case. And when I say high rates of T. cruzi infection, I mean more often than not. When we catch a bug in the wild here, it is 60 to 75% ish percent likely that it will have T. cruzi in it. So we average there around that 3/4 mark. Every season it fluctuates a little bit. And we are able to just molecularly test that it's T. cruzi, but we've also been able to pull out primary cultures from the bugs and establish them in laboratory culture and look at them, and they're definitely T. cruzi. So for veterinary health, so this is a lot about bugs and how to control them, but I know that I think I saw some people from Department of Health on here. And so we're going to just touch a little bit on veterinary health and human health, as far as the implications for why it's important for us to look at these, and some of the bigger problems that this parasite can cause. So more than 100 animals have been documented to be infected with T. cruzi naturally, or can be experimentally. Basically, it's any warm-blooded, non-avian vertebrate can house T. cruzi. So birds have a weird complement system, a blood complement system that readily lyses T. cruzi when it gets into their bloodstream. But all of us that aren't birds do not do that, and it just very efficiently establishes an infection in vertebrate animals. Pretty much anything that you stick it into, it will live. So on the right-hand side is a table. So these are wild and domestic animals in the Southeast that investigators here at Tulane and Georgia and Tennessee and Texas and some other places have looked, just basically done serosurveys or PCR of wild animal blood samples. And we get a lot of the same animals showing up. This is a truncated list. So this is not all of them that are all out there, though. Again, it will live in just about anything. But we have a lot of raccoons. I know some of the folks at Mosquito Control and here at Tulane were involved in a big raccoon study that one of the things that they looked at was T. cruzi, and it's here. Possums, same thing. Possums are another great harborer of Trypanosoma cruzi. The eastern wood rat and mice. This is a bias sample, but they do show up other places. Highly positive as well. This is a bias sample just because this is from that same stand of woods where we know that there was a human case and we get high bug rates, high positive bug rates. Then some of these other ones are things that you might think of less. So things like a coyote, 9-banded armadillo, and a bobcat. So wild animals like this, there are two other ways other than, I guess, two oral transmission routes that are poorly understood, but almost certainly are at play with sylvatic T. cruzi transmission. And so one of those is eating a bug. So you notice like 9-banded armadillo, like up to 40-ish almost percent of them had been found to have T. cruzi. But the amount of available bite surface on an armadillo for a bug that feeds on things that live in nests at night is not great. They don't have these great multi-generational houses. They're not super stable houses. They just kind of dig a hole, live for a while, and move on. And so it would be surprising that an armadillo would have so much of this parasite in it. But it is likely that they have it because they're insectivores. And so these bugs are out in the woods and they eat the bugs. So those highly infectious metacyclic trypanostigos that live in the rectum of the bug are also, remember, very easily transmitted across the intact mucosal membranes of the mouth and the esophagus and the GI tract. So basically, it just has to make it into your bloodstream before it hits stomach acid. And that's not hard for it to do. And we see oral transmission of T. cruzi like that in humans as well. There are a lot of outbreaks, not a lot, but there are large, significant outbreaks of Chagas disease in South America, especially with the fresh pressed juices, usually at some kind of fair or big event where they juice just like a whole bunch of acai berries or something like that. And a bug ends up mashed into the juice, or there's bug poop on the fruit and it's not washed before it's juiced. And in that way, we've seen events where hundreds of people at a single time are infected with T. cruzi. So it's a very, very efficient transmission method. The other way is that things like bobcats and coyotes probably get it from eating the rats or things like that that have high circulating parasitemias. So again, it's oral transmission. All you need it to be is blood to blood. If it's not in that highly infectious stage, if it's just in the regular trypanomastigote circulating blood stage or in a muscle stage, all it needs to do is be able to make it from blood to blood to continue that infection in the new host. And so a lot of these animals also will fight and draw blood between each other a good bit. And so we suspect that there's probably some horizontal transmission going on as well. And we see vertical transmission in humans from mother to child. And so it may actually be maintained in some of these animals in the absence of a vector being involved in any of it, which is how we think that there are things like raccoons that are urban raccoons that show up positive for T. cruzi. There's probably vertical and horizontal transmission between raccoons rather than involving a vector. The big thing for exotics and exotic animal rearers and hunters in this area. So just in the past couple of years, we've had a llama, a sloth, and an Asian small-clawed otter just between Louisiana and Mississippi that have all turned up positive from Chagas. And all three of these have been fatal in the animal. We have a host of rhesus macaques on the North Shore at the Tulane National Primate Center that are naturally infected, probably because they ate bugs that wandered into the colony. So again, pretty much anything can be infected. And with these exotics, whether it's that we don't have veterinarians that are just highly trained in watching them like a hawk to figure out what's going on, or if it's something about their actual physiology, but these infections have been pretty fulminant T. cruzi infections. They've died pretty suddenly. This was a big cause of concern. Folks in Texas found T. cruzi in white-tailed deer. We're still not quite sure how it got there. Presumably a deer ate a bug or bug poop on grass. But with things like raccoons and possums and white-tailed deer, these are game animals that people often field dress without gloves. And again, all you need is blood-to-blood contact. I know home in Mississippi, I'm not sure I've ever seen anybody wear gloves when they skin an animal from trapping or from a deer. And so this is just a really, really it's easy to see how efficiently this parasite might be being transmitted to small game hunters and hunters in this region. But we don't have any kind of serosurvey, and there's no medical monitoring that goes on for this population or is addressed towards this population, so we just have no idea. Canine Chagas is the other big one. Canine Chagas is more and more being realized as a real actual problem. Again, dogs eat bugs, dogs eat rats, or at least chew on them. And so there's a lot of ways that dogs could be getting this. There may be some cases if a dog sleeps outside in a dog kennel or, in a covered garage or something like that, there may be some vector transmission. But I know my dog just eats all the bugs that it can find just because it's curious. And you don't have to swallow it; you can just chew it up in your mouth. As long as you break the abdomen open, you can get those infectious stages right into your across your mucosal membranes. And so canine Chagas looks a lot like heartworm treatment failure. And so that's the big thing, is that if a veterinarian is not looking for it, if they're not testing for it, it often gets, often gets written up as a heartworm treatment failure, just because it looks like they died of a fulminant cardiomyopathy for no reason. And it can exist alongside heartworms. So you can absolutely have a dog that dies of congestive heart failure and cut it open and see a few worms and think, oh yeah, it was probably heartworm. But the pathology is actually caused by Chagas or a combination of the two. So there are people who are working on that and looking into not just diagnostics but treatments for dogs. Because one of the big areas that lost a lot of money on this was the U.S. military, who was training dogs for the armed services out in Texas, and they lost a lot of very valuable canines in their training school to canine Chagas. And so for human health, this was our big takeaway from the paper for human health. So 94.8% of the people, of the cases that were flagged by blood banks as confirmed T-Cruzi positive humans, 94.8% of those cases are not reflected in any public health system data anywhere. Not by the CDC, not by the states they reside in. No public health agency of any kind has any record that these people turned up positive. The American Association of Blood Banks, AABB, are the people who run the Chagas Biovigilance Network. It has recently, it was at least slated to shut down this year and stop reporting. So they're the only people who have been screening. It's a convenient sample. The blood supply is tested now for Chagas, almost at 100%. And all the AABB reporting blood banks do screen 100% of their units. And so these are people who turned up positive on initial screen, were traced, drawn again, and confirmed, and like have a person that is attached to that record confirm Chagas cases. And we don't know if these are locally acquired or imported. If they certainly a fair number of these people are immigrants from Chagas endemic countries in Central and South America, who also showed up in blood banking records. But we do know that a fair number of these are people with no travel history, who have never left, Central Louisiana or Central Mississippi, and have come up Chagas positive. And they report risk factors, just like you would expect, early childhood or, young adult, camping in the woods without a tent, low housing quality with no air conditioning as a child, those kind of things. There have been very few that we scratch our heads on and say, I have no idea where you might have picked this up. But this is of course because there are states that don't track this. It's not a reportable disease. It is here in Louisiana. It is supposed to be reportable, but even in Louisiana, we only had 87% of these cases that the AABB flagged were in the state's data set. Some of these states were more friendly than others with sharing data, and some of them were kind of hostile and dismissive of why we would even think that somebody should be keeping track of cases of Chagas in their community. I know that we do in Louisiana, because while it is not a highly communicable disease, there are implications for maternal and child health and for understanding the disease system and understanding, how it's working in the community and where certain populations might be more at risk or less at risk, and we can target interventions to help out the health of the state in that way. So there are places like Tennessee, they don't really report much. There's like somebody who had a post-it note at their health department that had the cases listed on it. They had like a file tucked away that wasn't part of the real file because they felt like it should be reported or at least kept track of. And we actually have a PhD student right now in the department who is spending all of her time combing through insurance records and hospital records to see if she can get a better count on how many Chagas cases are in these states and actually across the country, because we know that the AABB sample is a bias sample because not everybody donates blood. So these are just the people who feel like they were healthy enough and knew that they were, presumed to be healthy enough to donate blood and went in and turned out to be Chagas positive. So it was pretty striking that it was this many. Definitely led by Florida because Florida has 325 cases from the AABB surveys, and they don't keep track of it at all. So it is something that's definitely on the federal government's radar. They have a physician trainer based out of Texas that services Louisiana and Mississippi, physician educator trying to let physicians know that when they see things that look like Chagas, you should probably go ahead and test for Chagas, and not to ignore it because it is here. And it is something that even if you don't have travel history, you very well could be walking around with Chagas. So that is why we do all of this. And that is it.
Thank you, Sam. This was amazing. So I can see in the chat that there are a lot of questions for you. So I think I'm going to kick it over to Astacia so she can go through the questions.
Okay, the first question. Have you had many reports from Atlanta and North Georgia? I've heard of some.
Oh, you're on mute, Sam.
Yeah.
Sorry, I don't, thank you.
There you go.
I don't want to call it a cluster because that implies that it's not natural. But yes, there are cases up there. It's anywhere that's old growth hardwood. And it will push right up against the city. But yes, there are ones up there. There are several from Alabama. We know of locally acquired cases in South Carolina and North Carolina. Basically anywhere that someone looks for it, it's there. And so because of University of Georgia and the CDC, and a couple researchers up in North Georgia that are interested in this, I think that's why there are probably cases up there.
Okay. At a seminar, I heard about Chagas and trench fever being found in bedbug feces. Have you looked into this?
Yes. Well, we haven't looked into it. I've heard of this. So it's actually an experiment I wanted to replicate in the lab, and Dr. Weston said absolutely not, under no circumstances are you to have bed bugs in the lab. Which fair. So we don't know is the problem. Every time that somebody, at least with Chagas, every time somebody tries to run it through a bed bug, the experiments are not designed very well and leave a lot of room for critique. And so people don't want to believe it. Epidemiologically, it doesn't look like an issue. Again, if you have lots of times when someone in a house has Chagas, the other people in the house don't have Chagas. And no one's ever flagged a correlation with bed bugs. Nobody's ever been like, oh, well, they did have bed bugs in the house, and when they have bed bugs in the house, everybody in the house has Chagas. There's not those kind of correlations. So epidemiologically, it doesn't look like it's a pressing issue, but it may be a thing that does happen. And it's something that we're interested in because they are hemipterans and the parasite doesn't need to infect the gut. So it's possible that they survive the gut and get pooped out in their metacyclic form.
With mice being so high in prevalence, do we have to be worried about it being in their urine or feces?
No, it's the short answer. It doesn't. The parasite does not get passed in urine or feces. It stays in the bloodstream. It is preferentially attracted to innervated tissue. So, especially tissue that has nerves related to the parasympathetic nervous system. So that's why you end up with uh, parasites kind of focusing in the esophagus and the colon and in the heart. Those are the tissues that it's attracted to, and it never gets shed in any kind of way. It doesn't show up in saliva, urine, feces, anything like that.
Can humans become infected with T. cruzi by consuming infected venison?
It would be unlikely. It'd be highly, highly unlikely. And it would have to be like actively bleeding. One of the great things about T. cruzi from a pathogen, from a protecting human health, one of the, I guess, so a major weakness for T. cruzi from its perspective is that it's a protozoan, and it is easily killed by drying it out. So any kind of heat stress, any kind of moisture removal, it doesn't survive well at all. It's poorly transmissible. So just cook your venison and you'll be fine.
I've read articles about T. cruzi transmission from consuming fruit juice that had been contaminated with kissing bugs. Is this a concern in the US? And they said that they followed up that question with articles about this happening in other parts of the world.
It does. It does happen in other parts of the world. The only place that you would have problems with it in the US is if you were buying fruit juices from like a local fruit vendor in maybe the Southwest or in West Texas. And those fruit juices were like hand pressed at the farm and bottled, or you take right from the machine. Um, Anything in the US is going to be pasteurized and treated in some way that would make it impossible for T. cruzi to be passed in that way. The reason that it happens in other countries is that they don't have those kind of protections, and they have these big community fairs and gatherings around holidays that it is part of their tradition to take these fruits, make big vats of fruit juices, and then they just get distributed directly to a whole bunch of people, like a whole crowd of people. And that's a real recipe for disaster, because again, the infectious dose is so low that you just need one bug to get in there to effectively infect, a wide number of people, but not a risk in the US.
Here in California last spring, there was a yearling black bear that tested positive for Chagas. It was a first in California. Have you seen this in black bears before?
So we have not, but we also can't look at black bears very easily. I mean, it would be a great sample to look at, but the Mississippi River Valley's black bears are protected by and large. Um, and so interacting with them in any way is strongly prohibited. And we don't have connections with the people who do their blood draws, because I know that they do get knocked down periodically for samples. But the wildlife folks in the state are already stretched pretty thin. And so when we've approached them in the past about doing stuff related to Chagas and maybe doing like a Sierra survey of trappers, or getting involved with like the licensing of trappers, or looking at some of the bears or other animals that are out there, the answer that we get back is we would love to, but there aren't enough hours in the day. So they've been happy to answer questions, and they've been great. It's just they have limited bandwidth, giving their funding and personnel.
Could T. cruzi be transmitted vertically in humans?
It is transmitted vertically in humans. We know this to be the case. It is actually transmitted across the placenta. So it's not like some viral diseases where it's like transmitted around the time of birth kind of thing, and you can do a C-section and avoid a lot of it. It's not like that. The T. cruzi actually migrates across the placenta and infects the fetus during gestation. And it does cause a problem. But if the standard currently is testing cord blood in places where Chagas is endemic, so at the time of birth, they test cord blood, and if the baby is positive, they go ahead and treat the baby. So there are two medicines that we use for treating Chagas disease in humans, and both of them are highly effective for pediatric patients and for acute patients. So like some like 80+%, 90+% of babies do great and clear the parasite and go on to live normal lives if you catch it.
What do you suggest we need to be doing to get the message to pest control companies and residents?
That's a great question, because the bane of our existence is messaging, because about once a year, usually in August, the news gets slow and somebody around the state runs a doom and gloom kissing bugs are coming for you story on local news, and our phones start ringing off the hook. So better, more informed messaging from the get-go. I mean, I would love to see those pieces never run, because all they do is exacerbate and upset the population, and it's a problem, because the first news that you find, like the Wikipedia page for Triatoma sanguisuga, is just wrong from top to bottom. So there's all kinds of bad messaging that people and bad information that people hit as soon as you like Google anything for this pathogen system in particular. And so it's been a really hard time trying to talk people down. That's most of what we do, is talk people down from, hysteria by the time that they get to us. So if there are communications people out there, which is great because they land on the scientist people who aren't great communicators anyways, and we're the ones trying to talk them down. So if there are people who do communications who are interested in that, I would love any thoughts that you might have.
Okay. It was a pest control seminar, and he was recommending for us to wear proper PPE when dealing with bed bugs, like gloves during inspections, especially. Do you agree?
Oh yeah. So I mean, bed bugs probably aren't going to give you anything. But they're, they're probably full of the blood of the people who are around there. So yes, universal precautions are standard for most of these things. For kissing bugs, we never recommend that anybody touch them with a bare hand. When we do house surveys, house inspections for people who find them around their house, we recommend that people grab them with a Ziploc bag and turn it inside out if they're going to grab them. Most people aren't that brave. Most people will whack them with a shoe and then pick them up, or scoop them into a little bag or something. But yeah, I would not recommend grabbing any of these things by your bare hands, just because there's a lot that we know and there's a lot that we don't know. But the scary things to me are the ones where something shows up. Like years ago, there was a rat that was tested in New Orleans, and we found an old world hantavirus. Like, where did that come from? So we don't know what we don't know. So I don't think a pair of gloves is a lot to ask, especially since we're also used to it from COVID. I think masking and gloving is a lot more convenient now than it's ever been.
Where has the TC bat been found?
So it is mostly in Central America is where they found it. If you look around on, I think it's National Geographic has a great YouTube video. They're actually kissing bug species that live in caves, and they transmit it in their own little bat kissing bug cycle. So they're cave dwelling kissing bugs that go up and they feed on the bats while they're asleep. But yeah, we've looked for it here, or we want to look for it here. The samples that we have gotten from North America have so far been absent of TC bat. With white nose syndrome, it is in north of us. It is, of course, a concern that there might be some other pathogen in bats weakening their immune system. But everybody who's looked up in at least the US has not seen it.
Pest control comment. Since Sang is associated with old growth forests, there would be little pest control implications in suburban urban settings without the forest. Am I understanding that right?
So the yes. So is the short answer. So the people that we, at least in Mississippi, that I got complaints from and seem to be the loudest are the people who have like horses or like a little farm or a little homestead or live on a lot of several acres, like in the suburbs, pushed up next to a forest. Um, and they just get very surprised that there are things in their house or around their house that they're not used to, or they could kill their dog, or might wander in and bite somebody at some point. And they could make that group of folks that I had worked with previously seem to be very vocal about their displeasure for having those bugs around. In a urban area, there's next to no risk of ever seeing one of these bugs. There are no collection records from like New Orleans proper, and we've had people looking for these bugs in this like at Tulane since the 60s and 70s. So it has never shown up, and it is readily found right outside of town, still in the parish, but like as soon as you get into the wooded areas. And that's kind of a pattern that we see pretty much everywhere.
Okay, I'm not seeing any more questions in the chat, but you did get several comments thanking you for this informative webinar. And people were also happy to see what you were doing in the Weston Lab. So once again, thank you, Dr. Jameson, and thank you everyone for joining us. If you have any questions that you think of for Dr. Jameson, please email them to us at education@nola.gov, and we'll get them to him for you to get your answers. Please follow us on all our social media platforms. We're at NOLA Mosquito on Instagram, Twitter, and Facebook. And let me see, someone just put something else in the chat. Oh, that was Dr. Regal also saying thank you, Dr. Jameson. But once again, thank everyone for coming out today, and have a good afternoon.
Thank you, everybody.